Okay, so good morning, everyone. So we're going to start. Thanks a lot for attending today's webinar. So given that today's seminar is only online, the only option for you to ask questions is to write them in the Q&A tab, and don't hesitate to report any technical issue in the chat. Before starting, we have a small announcement, which is that you can still submit proposal for speakers for the spring semester until next week, Friday, and we will announce results mid-January. And with that, I'm happy to give the floor to Mike for presenting today's speaker.
Hi, everyone. Today I have the pleasure of introducing Tiffany Taylor. Tiffany Taylor completed her PhD in Evolutionary Ecology of Dispersal at the University of Oxford under the supervision of Angus Buckling. She then went on to do a postdoc in the University of Reading, after which she did a short university teaching fellowship, and she is now a Royal Society Dorothy Hodgkins Research Fellow in the Milner Centre for Evolution in Bath. So I'll give you the floor, Tiffany.
Thank you so much, and thanks for the introduction. I'm really sorry I can't be there. I would love to be, but it's actually quite hard to get to Montpellier from Bath. But I had the pleasure of meeting Michael when he came to visit us in Bath in May this year. So I'm really pleased to be able to talk to you all. So I'm going to be talking about how in my lab we use experimental evolution to unravel the genomic causes and evolutionary consequences of biased adaptive outcomes. So as you've heard, I'm currently a senior research fellow at the University of Bath. For those of you who don't know, Bath is in the southwest of England. It's not too far away from London, and it's the gateway to the beautiful southwest coastal path all around here. As a city itself, it's also rather beautiful, and it's also home, the University of Bath is home to the Milner Centre for Evolution, which is great because it creates a conversation between evolutionary biologists and all sorts of other biological sciences around the department, and it really feeds through a lot of the research here. So especially for myself, the interaction between evolutionary biologists and microbiologists has been really valuable. So if anybody wants to come and visit us and give a talk, please let me know, and I'm happy to arrange that. So yeah, I started my lab in 2016 after I got a fellowship from the Royal Society, and we've been interested sort of generally in the evolution of gene regulatory networks, but also more recently we've been thinking about processes that bias and constrain adaptive outcomes. And so these biased adaptive outcomes can be more predictable because there are fewer potential mutational spectrum revealed to selection, so that there are few options essentially. So these events can become more predictable because we can limit the amount of variation at the end. So what benefits are there to being able to predict evolution? Well, there was a nice example that came from the pandemic. There was anything nice to come from the pandemic. So this was a quote from Dido Harding, who is our health chief and head of the track and trace system in the UK up until May this year. And quite early on in the pandemic, she said that we've seen the virus mutate, we've seen the new variant emerge, which is something that none of us were able to predict. So the scientific community did quickly jump on this and said, of course, mutation was inevitable, and natural selection would select for those variants that had a higher transmission rate. So we could have surely predicted that new variants would emerge. But would we ever have been able to have the kind of predictive power that might allow us to anticipate which variants might emerge at the nucleotide level and cut them off at the pass as such? So what do we need to know in order to predict evolution? So we're quite good at understanding or predicting the fate of a known mutation within a population, but what do we need to know in order to try and anticipate which mutations might emerge? Well, firstly, we need to have a strong understanding of all the factors that are at play in determining adaptive outcomes. So of course, natural selection is going to be an important, a very strong factor, but it works in concert with other factors, such as mutational biases and other evolutionary constraints. So we need to know which factors are important. And then if we hope to be able to identify where and when these factors might be at play, for example, by looking at genome sequences, we need to have a very strong mechanistic understanding of how they work. We need to know what we're looking for. And ideally, we also need an experimental system that allows us to test the evolutionary consequences or the predicted evolutionary consequences of these factors that might be biasing or constraining adaptive outcomes. And I think that's where our recent work fits in quite nicely. So, we've been using experimental evolution to address questions such as these by trying to uncover genetic mechanisms that drive predictable evolution, and also understanding how they might constrain available adaptive routes in an evolving population. So we do this within the context of gene regulatory networks. So this is because they're really important for rapid evolution. So genes are frequently organized into groups where one gene will be responsible for switching on or off a group of genes. And this is more efficient than regulating the expression of each gene individually. So they're important for fitness. And a more formal definition might be that a gene regulatory network is a collection of molecular regulators that interact with each other and with other substances in the cell to govern gene expression. And this then determines the functionality of the cell. So these regulatory networks are a key fitness determinants as they enable an organism to adapt their phenotype to a changing environment. And we look at two-component systems, which are great because they provide a sense and response mechanism between the environment and the cell. And so these are really great in terms of sort of understanding how these gene regulatory networks evolve, because you can alter the signals that they respond to and understand the output of that change. So they're very common in bacteria, these two-component systems. And the way that they work is they respond to changes either internally or externally. This leads to a change of expression through regulatory elements, which changes expression of gene outputs, that leads to a change of phenotype. And there's lots of feedback between all of these. So if you look at an organism lifestyle, this tells you a lot about the fact that gene regulatory networks themselves are evolvable and adaptable. So organisms that live in very simple, predictable environments, for example, symbionts, will have very few gene regulatory networks, whereas those that live in complex environments and very changeable environments will have large, complex, and many gene regulatory networks. So we know they themselves are evolvable, but we don't know a lot about how gene regulatory networks themselves evolve. And we also know that they're key sites for evolutionary innovation. So for example, when an organism, and particularly I'm thinking about bacteria because that's what we work with, when they go through a niche transition, we find that these gene regulatory networks are very common sites for mutation. And that's because through a single mutation in a regulatory element, it can have a knock-on effect of many, many genes. So it can have very large phenotypic effects through just a single mutational event. And you see through these transition events, if you look sort of using bioinformatics, you frequently see that a number of mutations will be present in these regulatory regions, and these facilitate rapid adaptation. And that's really important when you go through a niche shift, because if you can't evolve rapidly, you're likely to go extinct. So we know that these gene regulatory networks are hotbeds for phenotypic evolution. And this is because phenotypes can evolve largely and rapidly through changes in gene regulation. And a good example is our own. It's not at all unique, it's just that we're human-centric and we like to think about evolution in our own terms. So this is looking back at a seminal paper back from 1975 by King and Wilson that was looking at the number of mutational differences between chimpanzees and humans. And if you just look at the number of differences, it's very hard to explain our phenotypic divergence between the two species. But if you look at where these mutations are, we can see that a number of them are within regulatory regions that are going to have an effect on lots of genes. And so this sort of helps to explain this phenotypic diversity. So we know that they're important for fitness and we know that they're important for evolution, but what do we know about how these gene regulatory networks themselves evolve and adapt? Well, there's still many questions remaining about about how they might adapt. And that's because there's this kind of paradox that exists between maintaining efficiency of a gene regulatory network so that your gene expression profiles are appropriate for the environment that you're in, but also revealing variation that selection can act on. And this variation at the gene regulatory network level comes from these non-cognate interactions. So these sort of interactions that aren't between a transcription factor and its target gene, but off-target genes. So it's this sort of balance that selection needs to maintain between insulating these gene regulatory networks, but in order to enable evolution and adaptability of these gene regulatory networks, this sort of non-target, non-cognate interactions can facilitate that process. So one way you might imagine a mechanism that allows expansion of gene regulatory networks whilst also maintaining specificity is through duplication and specialization. So this is a bit of a toy example, but this is showing in deep time. Perhaps you had a transcription factor that was able to initiate transcription of both genes 1 and 2, so they're co-regulated. But at some point in time, there's a random duplication event. And if there is selection for individual or decoupled regulation of these genes, what might happen is you get divergence between this duplicated transcription factor, such that over evolutionary time, you end up with 2 different transcription factors, one that specializes or that's able to interact and initiate transcription of gene 1, and another that's able to initiate transcription of gene 2. So a consequence of that process is built-in homology within these gene regulatory networks. So the ancestral state, you've got 1 transcription factor that's able to interact with both gene 1 and 2. And in the evolved state, you've got 2 transcription factors, each specialized to each interact with each gene. But selection is unlikely to be strong enough unless there's a really strong cost associated with these alternative interaction, or these ancestral interactions. It's unlikely to be strong enough to fully insulate this. And as such, this purple transcription factor may retain a weak binding affinity to sites upstream of gene 2 and vice versa. And if in some future environment, these weak interactions are actually beneficial, then selection can act upon those interactions, strengthen them, and actually it creates an opportunity for rewiring. And rewiring is a process where you change interactions between regulators and their target sites, leading to sort of new expression profiles. And it's this rewiring that's really important for these adaptive processes. So this built-in homology seems to facilitate opportunities for rewiring, but that's not because it's selected for, I don't think. It's because natural selection actually isn't strong enough to fully insulate these systems because of the way that they evolve and diverge. But actually in some situations that opportunity for crosstalk can create an advantage. So we look at this process of the evolution of gene regulatory network rewiring using an experimental evolution system, and our model organism of choice is Pseudomonas fluorescens. So this is a gram-negative rod-shaped bacteria. It was isolated from the leaf of a sugar beet plant in Oxford in 1989. So it's very well lab adapted and well enumerated. And it has a swimming flagella, or multiple swimming flagella, that it uses to swim through its environment. And if you put it on a motility agar plate, which is just a soft agar plate, the wild type motility looks something like this. And this is using a combination of both its swimming flagella, but also viscosin, which is like a biosurfactant, like an oily substance the bacteria can produce that reduces friction and allows it to sort of easily move over a surface. So the first thing that we did was to engineer a non-motile mutant. So the strain that we work with most of the time is SBW25, although I will be talking about other strains as I move through. So the first thing that we did with this strain SBW25 was to remove or to delete FLE-Q. So FLE-Q is the master regulator for the flagella genes. The flagella genes are organized in a hierarchy with FLE-Q sitting right at the top, and FLE-Q is required to initiate a transcription cascade. So if you remove FLE-Q, none of the other flagella genes are able to be switched on, even though they're all present in the genome. Okay, so that was the first step. And if you remove FLE-Q, you unmask this sort of something we call a spidery spreading phenotype. So this is a passive motility phenotype, just using that viscosin. So essentially what they're doing is just sliding over a surface using this biosurfactant. So then we also introduced a transposon into this viscosin gene, and that results in a completely non-motile engineered mutant. So what happens when you put this mutant onto the middle of an agar plate? So firstly, it does nothing, it doesn't move, but very rapidly, within about 48 hours, you see a slow spreading motility phenotype evolve, and then after about 96 hours, you see a fast spreading phenotype evolve, and you can see these by these sort of faster moving blebs emerging from this slow spreading zone here. And we can confirm that these are in fact producing or expressing flagella using electron microscopy and also transcriptomics. So they're able to overcome this engineered non-motile state very rapidly, and it wasn't clear to us how. So what we did is we sequenced up our swimming mutants, and what we found that there were just these first step motility phenotype was due to 1 point mutation, and the second step swimming motility phenotype was due to a second point mutation, and these were within the NTR-BC two-component system, and this system senses and responds to nitrogen in the environment and regulates nitrogen uptake. So I'm going to talk a bit about where those mutations were. So within the non-motile engineered version, the NTR system, or the nitrogen regulation system, is fully functional. This is what normally happens. So NTR-B is your histidine kinase, and this can phosphorylate or dephosphorylate NTR-C, which is the response regulator. So this is a two-component system, as I was talking about previously. So this responds, and this this senses, and this responds to the change in the environment. So NTR-C in its phosphorylated form can then go on to switch on nitrogen assimilation genes. But what happens in our first step, slow spreading mutant, is we found there was a single point mutation within NTR-B, that's the histidine kinase, in the PAS domain. And what this meant is that it no longer dephosphorylated NTR-C, or everything was happening in one direction, so it was leading to hyperphosphorylation of NTR-C, a buildup of phosphorylated or active NTR-C in the cell. A consequence of this was increased expression of nitrogen assimilation genes, so much so that it actually led to a toxic effect, because nitrogen needs to be regulated. It's good to have nitrogen in the cell, but not too much, so you need to regulate how much you have in the cell. And it also, and I'll get onto why later, it also initiated a weak interaction with expression of the flagella genes. And the second step mutation for this fast spreading mutant, we found a second point mutation within the helix-turn helix of the response regulator NTR-C. So you still have this hyperphosphorylation of NTR-C, but the result of this second point mutation was a reduction in the expression of the nitrogen assimilation gene, so slight compensation for this toxic effect, and increased expression of the flagella genes. So if you look at the transcriptomics, we can confirm that this was indeed what was going on. So on the left here, you've got genes associated with the bacterial type flagella, and on the right, you've got those associated with nitrogen compound transport, because obviously we're interested, because these mutations are happening within the nitrogen regulation system, we were interested in what was happening to genes there. So this first column is the wild type SPW25. These next two columns are just looking at what happens when you delete FLQ and then introduction of that transposon into viscosin. So these are the non-motile isolates. And then this is your slow spreading mutant is AR2S, and your fast spreading mutant is AR2F. And what we found with the bacterial type flagellum is what you might expect. When you knock out FLQ, you reduce expression of flagella genes. So I should say cold colors here are reduced expression, warm colors show increased expression. So you reduce expression of the flagellum associated genes, and in the slow spreading mutant you find partial upregulation of those genes, and the fast spreading mutant you find further upregulation of those genes, expression of those genes. And then looking at what happens to the nitrogen compound transport genes, again what you find is through deletion of FLQ you actually find a reduction in expression of a number of the nitrogen compound genes, which in itself is interesting. It might suggest that there's some underlying crosstalk there going on anyway. And through this first step mutation, it leads to increased expression of many of these genes. And that second step, fast spreading mutation, actually compensates for some of these. So you can see this sort of red block here goes to more orange, and that's true for a number of these blocks. You find slightly reduced expression in a number of them. And this is also confirmed through phenotypic analysis. So this is just looking at the y-axis, the doublings per hour, so the growth rate of these mutants across different nitrogen sources. So LB is a rich media with lots of nitrogen. And then we've got minimal medium where glutamine, glutamate, or ammonium are the only nitrogen sources available. And you can see that mutations within this nitrogen regulation pathway have pleiotropic effects. So normally when your NTR system is functional, ammonium is the preferred nitrogen source because it's cheap. You don't have to do any sort of post-modification to it. You can just utilize it fairly easily. But that's only true if you can regulate how much ammonium you can bring into the cell. So it's also more toxic in higher concentrations. So in that slow spreader mutant where you are no longer able to regulate how much nitrogen you bring in, you can see there's a large fitness cost associated with that when ammonium is the only nitrogen source, and that is slightly compensated for in the fast spreading mutants. So these are significantly different, showing a slight increase in growth rate with that second step mutation. So what has the nitrogen regulation pathway got to do with the flagella pathway, and how was it able to rescue its motility so rapidly and through so few mutations? Well, it became more obvious just by having a look at these proteins. So this is showing FLE-Q, the master regulator that we deleted, and this is NTRC, the response regulator, where we found those mutations that were conferring the motility phenotype. And hopefully just by looking at them, you can see that they are very similar. They share high structural homology. They are both sigma 54 dependent, so this is a sigma factor 54 dependent enhancement binding proteins, or EBPs. So these work in concert with the sigma factor to recruit polymerase to RNA polymerase to transcription, initiate transcription of their target genes. So they're both part of the same family. And in the absence of FLE-Q, NTRC is able to interact with FLE-Q binding sites. And you can see this, it's without any mutations actually in NTRC themselves. So that first step mutation just leads to lots of active NTRC around, or lots of phosphorylated NTRC in the cell. And that itself is enough to sort of allow these weak interactions with non-target binding sites. And then those second step mutations are all within NTRC that sort of reinforce that interaction. So you find, if you look at where the mutations are occurring in NTRC, they're all within this C-terminal DNA binding domain. So this is the domain that actually interacts with the DNA. And through some protein modeling, we were able to show that these mutations all make NTRC more FLE-Q-like, although this is quite a variable region, so it's hard to say that. But yeah, so it seems to be that these mutations are essentially, that first step is innovating NTRC, so it's creating opportunities for promiscuity or non-cognate interactions. And the second step refines, so it refines that interaction. So NTRC becomes better at interacting with FLE-Q target sites, and it actually becomes moves away from its own target sites, which is why you get that compensation for overactivity, because it's actually losing some affinity for its own binding target binding sites. So this was my postdoc work, and this is sort of what I brought to my own, start my own lab, because I sort of recognized that this was a really great model system to study gene regulatory network evolution. And that's because motility itself is a very easily observable phenotype. You can see it on a plate. And within this engineered mutant, it's only achieved by gene regulatory network rewiring. So as soon as we see a motile mutant, we know that rewiring has occurred, and we can look at the mutations that can conferring that. The evolutionary events are also really rapid. They're happening sort of within 2 to 5 days, and they're very repeatable. We know where these mutations are likely to occur within the NTR system. And we have a pretty good understanding of the genotype-phenotype relationship. So we can sort of understand these pleiotropic effects, and we can manipulate the nitrogen environment, for example, to understand how the selective environment also might feed back onto some of these dynamics. So this is where, so I started with my fellowship, and the first thing I wanted to do is keep it simple and sort of understand the mutational spectrum of that first step mutation, that slow spreading mutant. So what we did is we worked again with our non-motile engineered strain, and we let it evolve swimming motility, just by putting it in the middle of an agar plate and leaving it. We then sampled from the leading edge these motile mutants, and we sequenced them up to understand which mutations were conferring motility phenotype. So this is work that was led by James Horton, sort of makes up the bulk of his PhD, but he's still with me as a postdoc now. And if you're interested in sort of the meat of this project, it's published here. So I'm going to show you lots of graphs like this. I'm just going to take a minute to explain it. So the y-axis here is showing the proportion of different mutants that we recovered from those sequencing up those motile evolved isolates. This is in LB, which is a rich media, or M9, which is a minimal media where ammonium is the only nitrogen source. And each color corresponds to a different mutation type. So hopefully what you can see from this graph is that overwhelmingly a single mutation was dominating our mutational spectrum. And this was this A to C transversion at site 289 in gene NTRB. So parallel evolution, experimental evolution studies is common, and that's because we're working with very strong selection pressures and very simple selective environments usually. But parallel evolution to the nucleotide level to this extent is exceptionally rare. There was definitely something weird going on, and it captured James's imagination, and he became very curious as to what might be determining this very strong parallel evolution. And he came up with three hypotheses that I'm going to go through as I move through. So the first was that perhaps it had something to do with mutational accessibility. So this is kind of like mutational target size. So it basically describes how many mutations can confer the same phenotype. So if there's only one mutation that can confer a phenotype, then under selection for that phenotype, you're going to get parallel evolution by default. So the way that we tested whether or not there was only one or a few options to this motility phenotype is just we repeated this experiment a lot, and we did it across some different nutrient environments as well, just to see if that pleiotropic effect that we know that existed with these NTRB mutations might reveal some rarer mutations that we haven't uncovered yet. So again, we're looking at each color corresponds to a different mutation, and we're looking across LB, M9, so it's the high and the low nutrient environment, but then also with the addition of glutamine or glutamate. And again, what we found is that NTRB A289C dominated across all these different environments. It was robust across all these different environments. But we did find that there were other alternative mutations that were uncovered, that they're just more rare. So we could reject this hypothesis that it was just to do with the mutational accessibility, and there seemed to be something else going on that was really biasing the adaptive outcome of this to favor this single mutation. So the next hypothesis was that maybe it was something to do with clonal interference. So this describes when you have multiple clones arising in a population at the same time, they compete, and you would assume that under strong selection that the fittest mutation should win. So here, because we're interested in swimming phenotype, our measure of fitness is actually the strength of that swimming phenotype, which is measured by the relative migration area. So it's relative to the common mutant A289C. So that's why it's all centered around 0. So anything significantly above 0 is going to have a stronger swimming phenotype. Anything significantly below 0 is going to have a weaker swimming phenotype. And this is looking at all the different mutations that we'd uncovered so far in that previous experiment I just described. And this is across different nutrient environments again. So you've got a low nutrient and then addition of glutamate, glutamine, and high nutrient. And what we found is that one mutation was significantly a weaker phenotype, and that was this GLUN-K frame shift. So GLUN-K is also a gene within the nitrogen regulation pathway. It actually represses NTRB. So this was a weaker swimming phenotype, and that was true across all the different environments. But if you look at the other mutations, there was no significant difference in terms of the strength of swimming phenotype. And of particular interest was this mutant here, this A683C. So the reason this is interesting is because it's also an NTRB, it's also in the same domain, it's also an A to C transversion. So in terms of probability, you would expect it to be happening at a similar frequency, especially given that the strength of phenotype seems to be also similar. So it wasn't clear to us why we weren't seeing specifically this mutation at a higher frequency within our mutational spectrum. So this basically made us take a step back and think about the genesis of mutation. So there must have been something going on that meant that this A289C mutation was occurring first, at least before this A683C mutation. So I probably don't need to tell this audience, but just in case, mutation rate is not uniform across the bacterial genome. There are lots of things that can influence the rate of mutation at particular sites or particular locuses. And this isn't an exhaustive list, but it's just an example of how many factors can play a role, and actually can play combinatorial roles as well. But in particular, because of the localized region of this bias, so it's down to the single nucleotide, we were really interested in things that might have such a localized effect, things such as sequence variation that might really have these localized mutation bias effects. So at the same time, luckily, a PhD student, Louise Flanagan, who's just finishing up now, she was working with another Pseudomonas fluorescens strain, PF01. So this strain was isolated in Massachusetts a couple of years after SPW25. And she was running very similar experiments. So working with an engineered non-motile mutant, which we call PF02X, allowing them to evolve motility, and then sequencing up these motile variants to understand the mutations that were conferring this swimming phenotype. And what she saw was something quite different, a much more diverse mutational spectrum. And interestingly, she didn't uncover this common mutation, the NTRB A289C mutation de novo within this background strain, even though we did engineer it into this background strain, and it's perfectly fine. But interestingly, all these mutations are within the same nitrogen regulation pathway. They're all within the NTR system, and they're all leading to the same end result, which is a buildup of phosphorylated NTRC. So it's using the same mechanism, it's just using a much broader mutational spectrum, which is kind of what you more typically expect. But this allowed us the opportunity to compare these two strains and understand how the differences between them might be driving this difference in the mutational spectrum. So the first thing we did was just compare the sequences, nucleotide and amino acid sequences of NTRB. So the left here is showing a nucleotide alignment, and the right is showing an amino acid alignment. And you can see that there is very high similarity between these two strains at the NTRB gene. So we're seeing about 89% similarity, nucleotide similarity, and about 96% amino acid similarity between these two strains. So very high similarity. In red here, I've highlighted the commonly mutated nucleotide at site 289 or its corresponding amino acid threonine at site 97, sorry. And I've also in purple sort of shown the matches immediately up and downstream of these sites. So looking at the amino acid sequence, you can see that there's a perfect match between these two alignments, and there are no mismatches immediately surrounding this threonine. But looking at the nucleotide sequence, that's not true. So there are these 6 mismatches immediately up and downstream of this site 289. And so these are silent differences, and that's because a certain amount of redundancy exists in the genetic code where multiple triplicate codons will code for the same amino acids. So these differences do not translate to amino acids, but they are differences between these strains. And because we were interested in such a localized form of mutation bias, we became curious as to whether this small sequence variation, silent differences between these strains might be enough to drive that sort of strong almost deterministic mutation bias that we were seeing. So what James did is he engineered these 6 sites in AR2 on non-motile SPW25 strain to be like PF02X, and the same, he engineered these same 6 sites in PF02X to be like AR2. And so I've already shown you this data before. But what happens with the introductions of these 6 anonymous sites is a very different mutational spectrum. And he didn't actually uncover de novo this common A289C mutation within this background, although again, we did engineer it in, and it's perfectly viable and has the same strength of phenotype as it does in this background. So through these 6 synonymous changes, we were able to show that parallel evolution fell from more than 95% at site 289 to 0%, and it suggests that we were able to successfully break a mutational hotspot. And conversely, what happened in PF02X was through the introduction of these 6 synonymous sites, we found overwhelmingly across both LB and M9 that NTRB A289C was the most common mutation, and parallel evolution rose from 0% to 80% at that site. And this is exceptionally interesting because we say we hadn't seen this evolve de novo in this background before. So it seems to suggest that just through these 6 anonymous changes, we were able to successfully build the mutational hotspot. And this is really exciting because it makes us sort of question the role that synonymous differences might play in driving adaptive outcomes. So I want to move on to my second project now. So that was looking at how biases at the nucleotide level can really alter adaptive outcomes. And now I want to think about the gene regulatory network level. So at this point, we've done this experiment hundreds of times, and we've allowed rewiring to occur between FLEQ and NTRC. And we always saw innovation occurring exclusively through the NTR pathway. So it was always the mutations were in that NTR pathway and never anywhere else. But why is that? So I did explain, obviously, that there is high homology between these two transcription factors. So these are both transcription factors. So our first thing was, okay, is homology the answer? Is it just the most similar to FLEQ, and that's why it's recruited first? So both FLEQ and NTRC are part of a bigger family of transcription factors that are all sigma 54 dependent transcription factors. And you can see, so this is showing a predicted 3D structural homology tree, so showing you how similar these transcription factors are in terms of structure to each other. And NTRC is not the most homologous to FLEQ, so that can't be the whole story. There must be something else going on. And so this became the focus of PhD student Matt Shepherd. He's just finished, actually, he just did his Viva last week, and he's now a postdoc at the University of Manchester in the Brockhurst lab. And he engineered a double knockout mutant, so delta FLEQ and delta NTRC, so removing that commonly rewired NTRC transcription factor, so forcing it to go down another evolutionary pathway. And he left these to evolve in the middle of an agar plate. And the left-hand panel here is showing you that this is the time to emergence, so the time it took for these motile mutants to evolve. And this is in the single knockout mutant where just FLEQ is knocked out and NTRC is rewired. You can see that it takes about 3 days on average for these motility mutants to emerge, and 100% here shows that 100% of the independent lines evolved this phenotype within 6 weeks. So 6 weeks is the cutoff, because that's when the plates sort of become too dry and unusable. And on the right here, you're looking at a double knockout. So this is FLEQ NTRC knockout. So you can see that on average, these were taking about 2 weeks to evolve motility, so much longer, and only 9.4% of independent lines evolved motility. So this suggests that, firstly, excitingly, there are other routes available, but secondly, that they are much less accessible. And so our first step was to sequence up these evolved mutants and find out where these mutations that were conferring the motility phenotype were lying. And what we found in all cases was that there was a mutation in a histidine kinase. So again, another two-component system, histidine kinase of PFLU1131. So I'm sorry for the name. It's a putative two-component system. We don't know what it does. So the genes haven't been named. But PFLU stands for Pseudomonas fluorescens gene 1131. So that is the histidine kinase, and its partner is PFLU1132. That's the transcription factor. And reassuringly, this transcription factor sits within this same family of sigma 54 dependent transcription factors. So this suggests to us that, okay, it is rewiring through an alternative transcription factor, but this is also not the most homologous to FLEQ. If anything, it's even less homologous. So homology can't be explaining this pattern. So what else is going on to explain this evolutionary preference for NTRC? And so what Matt did is he compared this commonly utilized rewiring pathway with this alternative rewiring pathway to try and understand the differences and the similarities between them. So he came up with sort of 3 rules that determine whether or not there's opportunity for a transcription factor to be rewired. And I'm going to go through each of these again as I move through. So the first thing that was in common with both the common and the alternative rewiring pathway is that first steps were always within these histidine kinases. And the consequence of those was unlocking regulatory promiscuity of its cognate transcription factor. So I've sort of mentioned this, but I'll go over it again. So what happens with these two-component systems is that the histidine kinase will respond to an environmental signal or an internal intracellular signal, and under the correct signal, it will act to phosphorylate its paired transcription factor or response regulator, and its phosphorylated form, these can go and interact, these transcription factors can interact with their binding sites on the DNA. And what happens in all cases with these first step mutations is that it removes these ability of these histone kinases to respond to environmental cues, and it just drives interaction such that it's always phosphorylating its cognate transcription factor. So this leads to an increase concentration of active transcription factor in the cell. And once its target binding sites are saturated, that sort of leaves a potential for this active transcription factor to be loose in the cell, and it can interact with non-cognate targets. So if you look at the transcriptomic data, you can sort of see the consequence of that. So this first column here, you're looking at what happens in this double knockouts. This is delta FLQ, delta NTRC knockout. And the second column, you're looking at the gene expression profile and this common rewiring pathway. And in the third column, you're looking at the gene expression profile in the alternative rewiring pathway. And again, warm colors are high expression, low colors are low expression. So what happens in the flagellum genes might be what you expect. So this is, again, just looking at these slow spreading mutants. You can see that there's upregulation of the flagellum genes compared to the non-motile engineered mutant. In the nitrogen genes, you see in this NTR-BC rewiring pathway, a large increased expression of a number of the NTR genes. I've already talked about this. But you also see increased expression of the NTR genes within this alternative pathway. And this is a putative two-component system, but there's no reason to think that there should be any native interactions between this system and the NTR genes. And this story holds true if you look across all the other genes controlled by the sigma 54 dependent transcription factors. You find that in these, after this first step mutation, there's a large increase in expression in a number of these. And if you add it up, you can see that there's an increased expression for 70% of all of these genes controlled by sigma 54 dependent transcription factors in the NTRB mutant, and 54% in the 1131 mutant. So this first step mutation that leads to a buildup of phosphorylated transcription factor in the cell seems to unlock regulatory promiscuity and facilitate those non-cognate interactions that can lead to off-target expression effects. So far I've been talking about those first step slow spreading mutations, but I explained at the start of this talk that there's a two-step process. You get those slow spreading mutants, and then you get fast spreading mutants emerging from that. So these two circular plots are just showing, so the internal circle is supposed to represent the bacterial chromosome. Internal circle are these where the mutations lie on that first step mutation, and the outer circle is the second step mutation. The size of the dots corresponds to parallelism, so how many times we recovered the same mutation at that site. And this is showing the common NTR-BC pathway and then the alternative 1131, 1132 pathway. So within the NTR-BC pathway, we see this process, and I've sort of said this already, of innovation and refinement. So that first step mutation is basically leading to increased promiscuity of NTRC, and that second step is all within NTRC itself, refining that interaction of NTRC with its new target sites in the flagella pathway. And in both cases, they're highly parallel. I can actually add points. I haven't updated this, but there are more points here. This is just over because of the James's experiments. We have a lot of data for this first step, but we have more for the second step. I can confirm they're both parallel. But in this alternative pathway, you can see that first step is highly parallel. This is the mutations in PFLU 1131. But it's a very different story for the second steps compared to this NTR-BC pathway, where we see this large, diverse range of mutations all across the genome that seem to be doing very different things. So this created quite a big problem for Matt, trying to figure out what was going on in these second step mutations. How are these mutations all leading to increased motility in this alternative pathway? And he noticed that in all cases, and I haven't got time to go through all these mutations, but using different mechanisms, they would lead to increased gene expression of this hyperphosphorylated transcription factor. So after you've got that presence of that first step mutation, the second step is increasing expression, so making more of the active transcription factor. And there was a nice example from one of the mutants, which was a promoter capture event. So this is this first step mutation. where you see this mutation in PFLU1131 that leads to hyperphosphorylation of 1132. And then the second step is a 1.5 kb deletion immediately upstream of this mutation that removes a row-dependent terminator and sticks 1131 and 1132 under control of a predicted RPON or sigma 54 dependent binding site. And so what this means is that if you have lots of active 1132 around, it can then establish a positive feedback loop because it can bind to these sigma 54 dependent binding sites and drive its own expression. And you can see that if you look at the transcriptomic data, where the second step mutation leads to very large increase of expression of 1131 and 1132. And Matt was able to show the importance of expression in determining this motility phenotype by putting NTRC, this is that common rewired transcription factor, and 1132, that alternative rewired transcription factor, each of these independently under control of a rhamnose inducible promoter. So this is in the presence of that first step mutation. So there's already lots of phosphorylated transcription factor around. And what he showed is as you add rhamnose to the media, so as you increase expression of these hyperphosphorylated transcription factors, you increase motility. So it's showing you that high expression is important for that fast swimming phenotype. And interestingly, these two curves have very different shapes. And this is something that I'm really interested in and that we're sort of focusing on at the moment. So these open triangles are NTRC, the common rewiring pathway, and the closed circles are 1132, the alternative rewiring pathway. And it suggests that actually, if this alternative pathway is able to achieve high expression, it's actually more efficient at initiating this motility phenotype. So the line is steeper at the beginning, essentially. And this is probably to do with the number of binding sites. So it's easier to achieve that high saturation of its target binding sites sooner. But that's something that we're looking into more. But I was really surprised by this because it actually shows that if high expression was easier to achieve in this alternative pathway, it might actually be a more efficient way of rescuing your swimming phenotype. So why is it so much harder to achieve this high expression in 11312 versus NTRBC? Well, it becomes a bit more obvious if you look at the structure of these gene regulatory networks. So NTRBC has a native positive feedback built in. So what normally happens is NTRB, as I said, phosphorylates NTRC, is repressed by GLIN-K. But what these mutations do in NTRB is they essentially, or in GLIN-K, is they remove the ability of this repression. So what happens, that's how you sort of drives constant phosphorylation. And what happens is in the absence of this repression, you establish a positive feedback loop, and you can establish both hyperphosphorylation and hyperexpression through one point mutation. That is in comparison to the 1131, 1132 two-component system. Now, we don't know a lot about its structure because it's putative, but we can say that it doesn't have this built-in positive feedback. So one mutation can lead to hyperphosphorylation of 1132, but to achieve that high expression requires different mutations from around the genome because you don't have this positive feedback built in. So it requires two mutations to achieve that same phenotype rather than one in in the NTRBC pathway. And this is really interesting because it suggests that some transcription factors might be primed for rewiring of promiscuity just by virtue of the gene regulatory network that they sit within. So they may contribute to adaptation more readily than other transcription factors. And so together this work suggests that a transcription factor's evolution is constrained by the network it sits within. And using this knowledge, it might help us predict the likely adaptive outcomes of gene regulatory network rewiring. And this is something that we're trying to follow up further. I just really want to quickly mention how, because at the moment I've sort of talked a lot about quite artificial conditions. Of course, we're using experimental evolution and we're using quite contrived mutants to explore some of our questions. But we've been thinking a little bit about how crosstalk might facilitate gene regulatory network in more natural settings. And we wrote an opinion piece in Current Opinion in Micro earlier this year that sort of talks about how I think traditionally crosstalk is thought of as something quite negative, something that natural selection, of course, does act to insulate and it reduces crosstalk as much as possible. But we want to explore this idea of sort of, but does it create an opportunity for rapid adaptation as well? And so we discussed in this publication evidence for how these niche transition events might benefit from these crosstalk opportunities through either, we find that quite frequently you get mutations either in these histidine kinases that can lead to increased concentration of phosphorylated transcription factors that can create opportunities for promiscuity, but also how this built-in homology between transcription factors also creates opportunity for crosstalk. So it's just thinking about crosstalk in a slightly different way and how that might be important for rapid adaptation of bacteria in more natural settings as well. Okay, so I'm just going to wrap up now. So what are we doing in our lab? So we are identifying molecular factors that bias evolutionary outcomes. And so far, the picture that we're painting is one where it's a combination of genetic features, regulatory structure, and the environmental conditions that are going to significantly bias evolutionary outcomes. And by understanding all of those better, we might improve our predictive power. And we're also trying to use experimental evolution to understand the evolutionary consequences of these features for adaptive outcomes. So that might allow us to identify where and when strong bias is acting. Hopefully, the ultimate goal is by looking at genome sequences, although something that this work is bringing up is that it's actually really complicated, that you have to have a really good understanding of the mechanism to be able to understand where these processes are at play. And the end goal is sort of to predict, to improve our ability to predict evolutionary outcomes. I do have an opportunity for a competitively funded PhD opening in my lab at the moment with, I think, a January deadline. So if you know anybody that might be interested in this type of these type of questions, please point them in the direction of me and I'll put in touch. So this is a project working with a synthetic biologist in Bristol, asking whether or not some gene regulatory networks are more available than others. And finally, to acknowledge the people that actually did the work. So this is James Horton, Louise Flanagan, Matthew Shepherd, and Aidan Pearce, who I forgot to mention, but he was a brilliant research assistant that worked with us for years now, doing a PhD in London. The hotspot work was done in collaboration with Nick Priest and Rob Jackson, and this is the postdoc work that I presented at the start. So a really big collaborative effort. If you're interested in how this story develops, you can follow me on Twitter, and I'll happily take any questions. So thanks so much.
Thanks a lot, Tiffany, for the talk. So people can ask their questions in the Q&A tab, and while people can take time to type them, I think, Mike, you had a question yourself, right?
Yeah, I do have a question while people are typing. Tiffany, do you think it would be possible if we added a third mutation, or rather a third or fourth knockout, to completely prevent mobility coming back?
Yeah, that's a great question. We did, or Matt did that. He did create a triple knockout with FLEQ, NTRC, and 1132, and left it for 6 weeks, and we didn't find anything else was possible that rewired within that time frame and these selective conditions. So it's important to say that. But something else that Matt did that's really interesting is that the selective conditions are really important. So this idea of these two component systems rely on signals, right, from the external environment. So if the right signal is there, so you have quite an active regulatory network, then rewiring is more possible, because again, because it's active, it's being transcribed more, it's going to have a high mutation rate, and also these non-cognate interactions are going to be more likely to occur. So he has got also some work showing if you're just looking at this mutation, this sort of selective setup, then the limit seems to be, say, this alternative. There's the common and the alternative, but in the presence of the correct signals, you can get rewiring through these other transcription factors in that same family. But it depends on having the right selective environment. Does that answer your question?
Yeah, so you think there could be an environment out there that gives the right conditions?
Yeah, so
potentially.
it's about so you'd say it's the problem of having this opportunity for high expression is actually the hard thing. That's something that kind of came a little bit from this work, but you need to have the right signals to have that expression. If that's not there, then there's not, that variation doesn't exist. It's not revealed to natural selection. There needs to be the potential for that needs to be expressed in order to be able to be seen. So I think it's about, you have to really understand the selective environment that you've got these guys in to really, so there are, it's a really long way of saying there are alternatives, but you have to really tinker with the system to get at them.
Yeah, thank you.
Okay.
Just on the 6-week cutoff, do you think if you had a really long-lasting agar that after a year, or I don't know how long, that a mutation would pop up?
Maybe, although you are becoming a bit restrained. So at that point, it has used up pretty much all the nutrients in its environment. So it's going to be not dividing very fast. So there has to be some limit. I think at some point they're going to stop dividing, and then you're not going to see mutation anymore. So I guess there is a limit, but at the moment, yes, I think we are imposing a limit more because of the conditions of the plate just aren't appropriate anymore. I don't know. I guess perhaps I could try. I mean, the trouble is if you increase the humidity too much, so to get sort of stop the drying out process happening too quickly, then you can get condensation, and that will ruin, if you get any drops on the plate, then you know, the motility becomes obsolete. So it's a balancing act. So I don't know, maybe, but I haven't figured out the right conditions to do that yet. Although actually we are playing with static cultures at the moment. So that's something that perhaps we could run it for longer, because we do find that there is selection in just sort of using static liquid broth. There is selection for motility there. It's just not as strong. So you'd have to leave it even longer. So maybe.
Okay, thank you.
Thanks.
So we now have a question by Guillaume Cambrai.
Hi there. Thanks for the talk. I really enjoyed it. So as others, I think I have a very obvious question. That is, do you have an idea what is the mechanism behind the mutational hotspot that you actually observed?
Yeah, it's a good question. Do you know, and it's a really interesting question, because until recently I would have said, we've got, we basically know what it is, we're just ironing it out. And actually in the last sort of month, we've had to change our thinking a bit, which has been great. It's fun science when you get to do that. But so originally what we thought is that actually I've got a slide on this. We thought it was a hairpin structure, right? So the way that these hairpin structures form is you have these inverted repeats inverted complementary sequences on either side of a non-complementary bit. So you get during replication, when the DNA is single-stranded, what can happen is these complementary regions can come together and form a loop at the top of a hairpin structure. And our original thinking was that it could trip up the RNA, the DNA polymerase, and increase mutation at the base of these hairpins. And this is a known mechanism, and that's what we thought was going on. But James went in further, basically, and was trying to test this hypothesis by mutating the different sites within that hairpin structure. And we were able to show that actually that doesn't necessarily hold true, because there's only actually two mutations that are necessary to lead to this, to break or build this hotspot. It's actually not 6, it's 2, which is really surprising. So he does, we do have another theory, which we're working in collaboration with someone else who's noticed a phenomenon, a more general phenomenon, but I can't say too much about it because it's work in progress, and I know James is a little bit worried about scooping, but we think it's actually, importantly, it's a combinatorial effect as well. So it's not as simple as just the point mutations. It's also important as to where the gene is, what strand the gene is on, because something else we did, I think I also have a slide for that, so I've got all these hidden slides, but something else we did, this was published in MBE also last year, so to sort of test this idea of how what were the conditions required for this sort of strong hotspot. So is it determined by the location in the genome, essentially? And so to test that, what we did is we relocated the NTRB gene to a different site in the the chromosome, so we took it closer to the origin. We also flipped the gene and we moved it onto the alternative strand. So it was originally on the lagging strand, we moved it onto the leading strand. And obviously this is kind of quite a complicated figure, so I haven't got lots of time to go through it, but you can sort of see that as we mess about with the genome location of NTRB locus with this, either with the hotspot built in or with the synonymous mutations built in, we can gradually reduce the sort of parallel evolution at this site. So this all still holds true, even though it's probably not a hairpin we're talking about now. So it shows that there is an effect on, basically, the closer you are to the origin, you lose some of that parallel evolution, and that's because you have a higher concentration of DNTPs at the start compared to the end, so that leads to a lower mutation rate. Also, with it being on the lagging strand is important, because, well, actually at the time we thought it was because of the hairpin. That's probably not anymore, but the strand is important, and the orientation is important, and that's probably to do with collisions at that site. So it's really important. So basically what I'm saying is our original thought was, oh, okay, so now that we know that these, we can just look for these signatures of these sort of complementary sequences or whatever, and we can go into genomes and we can identify where these hotspots are at play, but it's really not that simple. And that's sort of why I was trying to hammer home that message of having a really strong mechanistic understanding is important if we want to be able to look at genome sequences and sort of say, we think this is a hotspot here. So I'd say that was a really long way of saying we are working on a mechanism, and I think probably early next year we will have actually proof of the mechanism, but it's to do with local nucleotide sequence. That's all we can say. Is that okay?
Thanks a lot, Tiffany. So we'll take a last question by Stephanie.
Hello, thanks a lot for your talk. So actually my question, can you hear me?
Yeah, I can now.
So my question was very similar to Guillaume's one, but I have another one. So I think the mechanism you describe has a lot to do with the fact that there has been duplications of this transcription factor of the mechanisms regulating the regulatory mechanisms. So are there equivalent of these regulatory mechanisms that are that haven't undergone duplications? I mean, are there modules in the genome that do not have this crosstalk phenomenon that we see as that would have less evolvability, and how could we explain this diversity in the systems?
Yeah, it's interesting. I haven't really looked at that, and I think the only way you could really get at that, and I'm sort of thinking on my feet here, is possibly looking at, say, for example, these symbionts that have very few gene regulatory networks, because when you work with anything, so for example, fluorescence that I work with, it obviously has loads and loads of gene regulatory networks, and the way that these evolve is through these sort of duplication specialization. So to find one that hasn't evolved through that process in something that's so diverse is probably not going to happen. And the other thing that's really hard when just sort of thinking how could you experimentally look at these is, and I sort of talked about that already, is you have to have an understanding of those strong selective conditions. So that's why motility is ideal, because it's something that you can really easily restrict and select for. So if you were to look at how the impacts of crosstalk, I mean, I guess something you could think about doing is some more clever engineering, is to really if you could engineer transcription factors and binding sites to be absolutely complementary, but then you'd have to also engineer all the other potential binding sites to not, but it would be really hard. I can't decide whether it would be easier to work with engineered strains or say symbionts or something. But actually, no, I think, I sort of mentioned this PhD that we're looking at hopefully getting someone to do, is using more a synthetic biology approach to sort of say, okay, so if we think that gene regulatory structure is important in determining what rewiring possibility, then we can create lots of different artificial synthetic structures, and we can plug in our different transcription factors from that same family, and basically say, well, we would predict that this transcription factor, this regulatory structure should allow rewiring with whatever transcription factor we plug into there. So I think perhaps using a synthetic approach, we might be able to get at questions such as that, but we aren't there yet. I think we're just starting to think about that at the moment.
Thank you.
Thanks. And last question by Ophélie.
Yeah, I just wanted to come back to your original question. So after all this work, would you say that we can or cannot predict evolution? And I mean, can we predict really a priori when molecular evolution will be highly convergent or not? Or can we just a posteriori try to understand when it's the case? Are we in the position now to like make really prediction about that?
Yeah, sure. I mean, I think it's being realistic. I mean, full predictive power is never going to be possible because it is, you know, mutation is a random stochastic event. So you're never going to have full predictive power. But I think in the two examples that I've shown, in both cases, there was one case, obviously, where you did have, you know, 95% of the time we can predict the nucleotide level which mutation is likely to emerge in our evolving populations, and at the network level, sort of a different level, to stepping back a bit, we can predict again within our original engineered mutants with 100% success that we know the rewiring pathway that it will recruit. So I think in some instances, and this is showing actually, especially with the rewiring work, I think that the number of options for rewiring are potentially really, really constrained, and that a number of factors will be working together, that there's kind of a Goldilocks spot about sort of which transcription factors are available for rewiring. So I think in these two instances, we can say we do have good predictive power as to how the adaptive outcomes that we expect to see. And I think through sort of stronger, if we can really understand the mechanism in this sort of model system, the idea is then we can hopefully use that knowledge to understand how where you know instances of bias and constraints in other organisms. And also, interestingly, we might be able to use this knowledge to better design, and so I keep talking back to synthetic biology, but better design synthetic organisms to kind of have that element of predictability, because you need to know, especially with synthetic organisms that have the potential to adapt, you want to know where they're likely to end up in order to, if they're going to keep doing the thing that you need them to do. So I think, like I say, full predictive power, no, but lots of lessons I think can come from this work that can allow us to anticipate adaptive outcomes under certain scenarios. Is that okay?
Okay, thank you.
Thank you.
Thanks very much. I think we can stop here.
Thank you.
Thanks very much, Tiffany.